Good afternoon, everyone, and welcome to today's Biomarkers Series webinar, Dynamic Biomarkers for Deep Immune Phenotyping in Autoimmune Disease. My name is Esther, and on behalf of Oxford Global and Sapient, I'd like to thank you all for joining us today. After the presentation is complete, we will have a 10-minute Q&A session. Please submit your questions through the question panel on your menu display. The full recording of this webinar will be made available to download on our secured website and to shortly after today's session. All of today's webinar, all attendees have been entered into a prize draw to win a Plus Pass from Oxford Global. The winner will be contacted directly with instructions on how to claim the prize on Monday, the 9th of October. Autoimmune diseases present with a wide range of clinical symptoms, severity, and therapeutic response, complicating the diagnosis and treatment of patients. In this webinar, Dr. Mo Jain and Dr. Tanya Nguyen will discuss how high-throughput mass spectrometry-based methods can be applied to broadly assay thousands of small molecule biomarkers, which read out dynamic changes that occur with immune system activation, dysregulation, and response to drug exposure. They will share how these biomarkers may be leveraged to deeply phenotype the immune system and even classify autoimmune disease subtypes, monitor for therapy-related immune adverse events, and predict response to targeted therapies. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, computational biology, and mass spectrometry-based bioanalytics, and is founder and CEO of Sapient. Dr. Nguyen is a clinician scientist with over 13 years of research experience, leveraging multi-omics discovery within the context of human biosampling, and is head of scientific operations at Sapient. Without further ado, please enjoy the webinar, and over to you now, Mo and Tanya.
Thank you very much, Esther. And we're very excited to be here today on behalf of Sapient to talk about our work to understand dynamic biomarkers for deep immune phenotyping in the setting of autoimmune disease. Now, if any of you that work in this area are well aware, there represents tremendous heterogeneity, both in the clinical outcomes as well as in the phenotypic spectrum of autoimmune disorders. Not only the underlying organ system that can be affected, but the gender predominance, the age of onset, and even the frequency, which can range from certain disorders such as psoriasis that occur in more than 1 in 100 individuals to very rare forms of autoimmune disease that occur in less than 1 in a million individuals. Now, despite this great heterogeneity, there seems to be a predominant phenotype in which autoimmune disorders continue to increase annually, both in the incidence and in their worldwide prevalence. Now, much of this heterogeneity is due to the evolving landscape of disease etiology. Now, while we often think of these autoimmune disorders as being heavily genetic in origin, what we find is that when we look across concordant twins, or those twins that share 100% of their underlying genetic material, that there still remains quite a bit of discrepancy in disease etiology, suggesting that autoimmune disease is highly multifactorial in origin. Germline genetic variation accounts for less than 20% of total autoimmune disease trait variability, suggesting that other non-genetic factors, whether they be non-coding mutations that modulate gene expression, autoantibody formation, epigenetic reprogramming, and environmental factors such as exposures, toxicants, microbiome, diet, etc., can have tremendous influence in the development and phenotypic heterogeneity of these disorders. Now, this heterogeneity is owing not only to canonical autoimmune disease, but also evolving therapy-related adverse immune events. We know in the setting of cancer immunotherapy, severe immune-related adverse events are present in greater than 40% of individuals with various solid tumors. In over 30% of individuals with pre-existing autoimmune disease, there is a noted flare that oftentimes occur under immuno-oncology treatment regimens. Similarly, outside of the oncology space, with gene and cell therapies, there's a noted immune event that oftentimes is present, both owing to pre-existing immunity related to NAbs, T-cell immunity, disease-associated immunity infections, as well as post-therapy induction of the immune system that can have important influences on systemic inflammation and autoimmune events. Now, this great heterogeneity has important implications, not only in the diagnosis of disease and understanding long-term prognosis, but also, most importantly, in the treatment of these disorders. As well, as many of you are well aware, there's a lack of a universal drug response, with present therapies largely acting in a broad manner in a non-disease specific way that allows for limited overall efficacy. Up to 30% of patients treated with anti-TNF therapy in the setting of autoimmune disease actually do not respond initially. And even among those individuals that have an initial response to autoimmune-based therapy, up to 80% of patients will discontinue their autoimmune therapy over time, not to de-escalation and response, but rather to a lack of response and potential side effects that originate over time. Now, given this underlying heterogeneity, it is critical that we provide subclassification in a way that allows us to be able to understand subtypes of individuals, not necessarily around the host, but specific subtypes of the disease that have different diagnoses and different prognosis, and once again, different responses to particular therapies. This is where biomarker-guided drug development can be absolutely instrumental, in a manner that allows us to take the various forms of disease pathobiology and align them within specific people and in specific therapies. We could start with individuals that have a shared autoimmune diagnosis, and using biomarkers as companion diagnostics, begin to subset individuals and their disease processes into specific disease pathophysiologies or disease pathways that are altered. In this way, we can isolate individuals that are most likely to respond to a specific therapeutic. Now, when we think of these biomarkers that guide therapy, there's many different modalities, and it's particularly important in the setting of autoimmune disease to consider and think through small molecule biomarkers. Today, we have Dr. Nguyen, who's going to walk us through applications of small molecule biomarkers, specifically within the goal of autoimmune disease.
Thank you so much. And so these small molecule biomarkers provide a readout of the genetic and non-genetic factors that contribute to human disease. These are organic metabolites and lipids that come from endogenous and exogenous sources. And these molecules are special in that they are produced intracellularly. And because of their physical and chemical properties, they're released into the blood where we can capture and measure them. These molecules are highly dynamic, allowing us to monitor disease progression, drug response, and patient heterogeneity, oftentimes in real time. And lastly, these small molecules are highly conserved across species and models, from cells in a dish to rodents and non-human primates, all the way to human studies, allowing us to identify biomarkers that can translate across different species and models. Now, when we consider the full chemical diversity of small molecules in the human body, traditional mass spectrometry approaches have only focused on a small subset of molecules that are highly abundant. However, these represent only about 5% of the molecules that are found in the human body. However, using non-targeted discovery mass spectrometry approaches that we have developed here at Sapient, we are able to not only measure the 5% of known molecules, but we can also examine the full breadth of chemical diversity, which could span beyond 75,000 molecules circulating in human blood. Here at Sapient, we have made great technical advancements in the capture and measurement of these small molecule biomarkers using our next generation rapid liquid chromatography mass spectrometry systems, or RLC for short. Here's a brief video of our discovery lab located in San Diego, California. These high-throughput systems allow us to take any type of biological sample from human, preclinical, or cellular systems, and we can analyze biofluids such as plasma, serum, CSF, urine, all the way through various types of tissues, as well as cells and media. And very rapidly, we can assay upwards of 15,000 small molecule metabolites and lipids in a single sample. And we do this very quickly. Our analytical cycle time for analysis of one sample is less than a minute. And given that we have fully automated and robotic systems, we have enormous capacity to analyze several thousand samples per day. In each of these biological samples, we can readily identify approximately 700 molecules up front using our commercial standards library. With the remaining unknown molecules, these are characterized using what we call 4D chemical characterization. That includes retention time on column, accurate mass MS1, MS2 chemical fingerprint, as well as CCS on ion mobility. And these 4D fingerprint allow us to label each unique unknown molecule and eventually isolate that molecule for identification as needed. All of our projects and data generated on these systems undergo rigorous quality control monitoring and analysis that ensure instrument performance, sample integrity, and many other parameters that are related to data quality. Lastly, we've also leveraged these same mass spectrometry systems to perform discovery proteomics, as well as high sensitivity cytokine multiplexing that can be performed on the same sample aliquot. So given the high throughput nature of our mass spectrometry system, this enables population scale discovery. And so as part of Sapiens development, we have already analyzed over 100,000 unique biological samples that have been collected from around the world. These samples have already been analyzed using our rapid LCMS systems, measuring tens of thousands of molecules in each sample. And all of that mass spec data, alongside the clinical data associated with these samples, have been amalgamated into the central database that now contains over 130 million phenotypic data points. This database contains everything from basic demographics, rich phenotype information related to health and disease states, as well as environmental and lifestyle factors such as diet and exercise. Most importantly, the database also contains longitudinal clinical outcomes that have been adjudicated across a number of therapeutic areas and have been enriched for specific autoimmune and inflammatory diseases of interest, such as inflammatory bowel disease, rheumatoid arthritis, psoriasis, asthma, lupus, etc. It also contains many other diseases where immunity and inflammation have been implicated, including cardiovascular disease, metabolic, liver diseases, as well as rare diseases. And lastly, this database also includes matched genetics information, as well as a whole host of other factors that are associated with inflammation and immunity that are related to different biological processes and disease states, such as immunotherapy, response to immunotherapy in the context of other diseases like cancer, the microbiome, which has been implicated in immunity, and we have matched microbiome and metabolomic measurements in thousands of individuals. We also have measurement of other immune markers, such as cytokine levels and immune cellularity, as well as other disease states, such as acute infection, response to vaccine, and other diseases that I've mentioned thus far that have been implicated in immunity. And lastly, we also have dense serial sampling. If you can go back to the last slide. We have dense serial sampling measurement from individuals every hour over the course of a day, every day over the course of a week, and during various fasted and fed states. And all of this information allows us to understand how a molecule may behave under different conditions and how that may be influenced by different circadian and diurnal rhythms. As part of our discovery process here at Sapiens, we can perform multidimensional data analysis and integrate complex data sets. And this is particularly important in understanding immunity and inflammation. As mentioned, in our human biology database, we have 20,000 individuals in whom we have dense genotyping, in addition to corresponding blood samples that have already been analyzed on our mass spectrometry systems. And by leveraging genome-wide association studies, we can identify associations and map small molecules in circulation to specific genetic variants in the genome. So across these individuals, there are over 66,000 circulating small molecule biomarkers that have been assayed. For nearly half of the markers in human blood, we can identify a gene variant that is associated with a particular molecule at a genome-wide threshold. And this is a very powerful tool that once we can identify a biomarker, it allows us to understand the biosynthetic origins of that particular marker. We can also go in the opposite direction and use the genome as a starting point. We can take genetic variants that have been implicated in autoimmune diseases and identify specific small molecules in circulation that provide a readout of that gene. And so when we've looked across the 20 million or so common genetic polymorphisms, over 5% of them associate with a small molecule in blood. This percentage is even greater when we enrich for those common polymorphisms that occur within the coding or transcribed regions of a gene. These are the common polymorphisms that are believed to alter protein function. And across the 20,000 genes in the human genome, over 90% of these have a circulating small molecule biomarker in blood that are associated with them. So this allows us to identify markers that read out the function of a particular gene or protein, which is really instrumental in measuring and monitoring target engagement. So Sapiens technology and platform are fundamentally tools that can be applied to answer a number of different biological questions related to autoimmunity and inflammation, including discovery of novel markers that can classify autoimmune disease subtypes, predict response to targeted therapies, as well as monitor for therapy-related immune adverse events. And to quickly summarize Sapiens' capabilities, we are a purpose-built CRO that aims to provide state-of-the-art bespoke services to discover various types of dynamic markers. These include everything from metabolomics and lipidomics, discovery proteomics, as well as targeted protein panels, cytokines and chemokines, as well as providing NGS and other circulating DNA sequencing. In addition to the bioanalytical capabilities, we also provide advanced bioinformatics and integrative analysis that incorporate multi-omic targeted and non-targeted data. We can provide cross-validation of such discoveries by leveraging our in-house longitudinal human biology database. And we also have a CLIA-certified, CAP-accredited clinical laboratory that allows for rapid translation and clinical implementation of new discoveries. And lastly, our goal is to deliver data faster to allow for more rapid time to insight, with less than 60 days from sample receipt to results.
Thank you very much, Tanya. And I'm hoping in the last several minutes here, we can take a little bit of an adventure and walk through some actual real world data by which we can leverage these tools to understand both the immune function as well as autoimmunity, particularly within the setting of immuno-oncology. Now, this is a program that was initiated together with a collaborator, Sonia Sharma, and her laboratory at the Lowy Institute of Allergy and Immunology. As many of you aware, the immune checkpoint blockade has really transformed solid tumor therapeutics, in which various therapeutics can be leveraged to reawaken the immune system in order to target tumor cells. Now, as you can imagine, there's tremendous heterogeneity in the response to these various therapeutics, with some individuals having a remarkable response and others having a minimal response to these therapies. In addition, by reawakening the immune system, individuals are prone now to autoimmune adverse events. And so there's been great interest here at Sapiens in being able to leverage our tools to understand not only those individuals who are most likely to respond to these therapies, but also to be able to denote and substratify those individuals that are likely to have an autoimmune event in the setting of immune checkpoint blockade-based therapies. To begin with, we were quite excited about understanding particular biomarkers in circulation that read out the PD-1 pathway and made to note those individuals who are more likely to respond to a particular therapeutic in this space relative to folks who are likely to not respond. To begin to initiate this program, we looked at those common genetic variants occurring in the proteins that are central to the PD-1 pathway. And when we look in Sapiens Human Biological Database across the tens of thousands of circulating biomarkers in relation to common genetic variants in the PD-1 pathway, we find that there's over half a dozen molecules, as shown here in red, in circulation that are highly specific and statistically associated with changes in the PD-1 pathway. Now, one of these markers circled in blue here is particularly interesting to us as it appears to be very specific for the actual PD-1 pathway and reads out those individuals in which there's higher versus lower overall PD-1 pathway activity. If we look at this one singular biomarker now in individuals that are undergoing PD-1 based therapy, what we find is relative to pre-treatment, this marker acutely increases in the post-treatment stage. Now, even among these individuals, you can see that there's an underlying heterogeneity in which post-treatment, some individuals have a very high level of this marker, suggesting very high levels of PD-1 activity, whereas other individuals have a more subtle elevation in this one specific marker. If we substratify individuals relative to this single biomarker that's reading out PD-1 total activity, what we find is that this has very important implications for actual response rate. So those individuals that are at the highest quartile for response, meaning that they have the highest level of induction of this PD-1 biomarker, actually are associated with a 50% increase in response to these overall therapeutics relative to those individuals that are in the bottom quartile for response to these actual biomarkers that denote the PD-1 pathway activity. In this way, we can begin to not only identify biomarkers of this immune-related activity, but also to leverage these biomarkers now to substratify individuals according to those who are most likely to respond to a particular therapeutic in this space. Similarly, we can use these techniques to understand markers that denote individuals who are likely to have an adverse event, particularly an adverse autoimmune event in the setting of immune checkpoint blockade. This program was initiated across several human studies, both discovery and validation, as well as preclinical models in which we had autoimmune measures. These biological samples were analyzed using our high-throughput RLC-MS systems with the generation of this non-targeted mass spectrometry data that underwent rigorous biostatistical analysis to identify candidate autoimmune metabolites that are associated with the development of severe immune-related adverse events in the setting of checkpoint blockade. Now, to begin with, we initiated this study looking at a particular clinical trial of one such agent targeting the CTLA-4 pathway. And when we look across these biological samples, again, assaying tens of thousands of these circulating biomarkers as shown on the volcano plot on the left, we can begin to statistically ask, after adjusting for multiplicity, are there specific markers in blood that denote those individuals who are likely to have a severe immune-related adverse event in the setting of immune checkpoint blockade? What we find is that one such molecule, as circled in red, or as denoted in red and circled in blue here, is particularly altered in those individuals that are developing autoimmunity in the setting of these various therapeutics. Now, if we look at the change in this one molecule over time in individuals, what we find is that those individuals who are free from adverse events actually have flat levels of this molecule. Similarly, those individuals that develop grade 1 or 2 mild autoimmune disease, such as mild colitis or mild dermatitis in the setting of these therapeutics, similarly have a very flat level of these markers. Those individuals, however, that develop severe grade 3 or grade 4 autoimmune events, particularly dose-limiting events that can be life-threatening, we find that there's an acute drop in these biomarkers that precede the actual clinical development of autoimmunity. In this way, this singular biomarker may be leveraged to diagnose individuals at a very early time point who may be prone to developing of autoimmune events in the setting, once again, of these immuno-oncology-based therapeutics. Now, this was a discovery that was made in one cohort when we'd be able to thankfully extend it to various other therapies and various other studies of immune checkpoint blockade for solid tumor therapeutics. We can see across 3 independent studies from 3 very different places in the world and very different populations of solid tumors, there's a similar phenotype by which this singular biomarker is decreasing in those individuals that develop severe immune-related adverse events. Across a meta-analysis, we can see the log odds ratio is consistent, denoting or suggesting a universal response to various therapeutics in this space. Now, we can use Sapiens Human Biological Database to look at normal individuals who are free from solid tumors and ask similarly, how does this singular biomarker map onto the development of canonical autoimmune disease over time? What we find is that across more than 10,000 individuals followed for over 10 years, low levels of this marker are associated with increased level of canonical autoimmune disease, whether it be lupus, RA, psoriatic arthritis, or similar disorders, as shown in the Kaplan-Meier plot to the right. Now, we spent quite a bit of time understanding the underlying biology of this single biomarker and how it may be reading out individuals who are prone to developing severe immune-related adverse events in the setting of immuno-oncology agents. What we find across normal individuals in which we have immune cellularity measures is that this one singular biomarker is very much related to circulating levels of a particular subtype of neutrophils, as denoted in the image on the top upper left. Across these individuals in large populations, as would be expected, we find that the biomarker is very much related to circulating neutrophils, in that those individuals that have lower level of the biomarker, as shown on the image on the bottom left, have elevated levels of neutrophils. And we can see through the quartile analysis, as this biomarker level increases, that the level of neutrophils decrease. Now, if we look at these specific subpopulations of neutrophils in individuals undergoing immuno-oncology-based therapeutics, what we find over time, as perhaps would be expected, is that these populations of immune cells are specifically increasing in individuals, particularly those individuals that are developing severe grade 3 or grade 4 autoimmune events in the setting of these therapies. Now, this is a marker, given its underlying conservation, as Tanya alluded to earlier, that we can also begin to evaluate and validate in preclinical systems. We've examined 2 preclinical systems, a humanized CTLA-4 mouse that when upon receiving CTLA-4 and PD-1-based therapeutics develops an autoimmune colitis, as well as the DSS colitis mouse model that many of you are familiar with. To begin with, on the bottom left here, in the humanized CTLA-4 mouse, upon induction with CTLA-4 and PD-1 therapeutics, there's an acute colitis, and similar to as observed in humans, we can see an acute drop in this one single autoimmune-based biomarker. When we look across the spectrum of preclinical samples in the middle image, what we find is the biomarker is very much related to the underlying degree of toxicity, with those preclinical systems having the lowest level of biomarker being associated with the greatest degree of toxicity. Similarly, in the DSS colitis model that has a component of autoimmunity, what we find is with induction of the colitis, there's a similar decrease in overall biomarkers, as is phenotyped in the humans and the CTLA-4 mouse. Now, interestingly enough, these approaches allow us to discover new correlated biomarkers that may denote individuals at high risk. But in this case, we are also understanding biology that has causal relation to actual autoimmune toxicity. So as we've seen in the CTLA-4 mouse, there's an acute drop in this marker that's associated with acute toxicity, and repleting this one biomarker in circulation through IP injection of the molecule actually rescues the underlying toxicity that's seen by the histology score on the bottom left. Similarly, in the DS colitis model, rescuing the levels of this one single molecule results in a rescue of the underlying autoimmune colitis as marked by the histology score. Now, these changes in the actual histology metrics around toxicity are similarly associated with drops in circulating neutrophils upon repletion of the singular biomarker, suggesting a causal relation, once again, between this one unique biomarker, the development of autoimmune events, and neutrophilia. I'm going to go ahead and stop there. I would really like to thank the audience and time allotting. We'd be happy to take any questions.
And if you're interested in learning more, please reach out via email or the QR code that's shown here. Also, we'll be presenting at the Biomarkers US conference later this month on October 12th and 13th. Our talk is entitled Dynamic Small Molecule Biomarkers to Improve Patient Selection in Clinical Trials. We hope to see you there, and please stop by and visit us at booth number 3. Thank you.
Great, thank you so much, Mo and Tanya. I would now like to open the floor for discussion. Please remember to send us your questions using the question panel on the platform. So our first question is, can you describe the current landscape of precision-guided autoimmune therapies?
Absolutely, Esther. Happy to take this one and talk through this. It's a great question. And as you're well aware, this is an area that's rapidly evolving. Many of us know of the recent Prometheus story, for instance, that's received quite a bit of press, and their incredible success story in using precision-guided approaches in inflammatory bowel disease, and how this enabled them to go from R&D to target identification to novel biologic to a blockbuster phase 2 study that was highly successful in absolute record time. And this is a story that, while has played out in several areas, is going to become more the norm rather than the exception over time. And there's a number of similar stories that are evolving that have not yet been reported, but are in the evolutionary process for both RA, psoriasis, lupus, and immuno-oncology-based therapeutics. And all of these are leveraging biomarkers as part of the development process for more precision-guided approaches, whether it be for understanding targets, as we discussed, or for substratifying individuals into those that are most likely to respond to a particular therapeutic. Now, as a whole, this space has been heavily driven by oncology over the last decade or so, particularly with the advent and applications of parallel sequencing. But immunity inflammation is really poised to be the next therapeutic area, and this includes autoimmunity, of course, that is likely to be transformed by biomarker or precision-guided drug development. So while we're still in the early days, this evolution and this landscape is certainly growing and changing very, very quickly, and we're excited to watch this occur.
Great, thanks, Mo. So the next question is, how do small molecule biomarkers compare with protein and genetic markers of autoimmunity?
I can take that question. So small molecules absolutely provide complementary information to traditional genetic and protein biomarkers for autoimmunity. And it's important to recognize that small molecules aren't just providing more of the same information, but they fundamentally capture different types of information and have different properties. For example, as you're mostly aware, that the genome is static from the moment of conception, and it doesn't change throughout one's lifetime. But these small molecule biomarkers are highly dynamic, and they provide a readout of what's actually happening in real time with health and disease status, with different types of environmental exposures, diet, lifestyle, etc. Relative to proteins, there are important differences with small molecules as well. So as you know, proteins are made intracellularly, and it's quite difficult and bioenergetically unfavorable for a cell to export proteins into circulation unless cells die and rupture. And because of this, only a small fraction of intracellular proteins are actually found in circulation. On the other hand, small molecules are made intracellularly and then are rapidly transported via transporters across the cell membrane into circulation. So the vast majority of small molecules that are made within the cell are meant to be found in circulation and where we can measure them. Proteins also come from endogenous sources, whereas small molecules can come both endogenously and exogenously. And they reflect a combination of influences that not only occur within the body due to health or disease, but they also reflect changes that are due to therapeutic intervention, environmental factors, the microbiome. And so while traditionally we have focused on the measurement of these small molecules, we're also we also have capabilities for doing large-scale genomic measurements, as well as a range of proteomic measurements, to be able to integrate all of these types of measurements in better understanding biology as well as drug effects.
Great, thanks, Tanya. So the next question is, the concept of autoimmune disease subtyping is interesting. Can you further explain how you think about using your technology for this?
Sure, Esther. Happy to expand upon this. We oftentimes think of these autoimmune disorders as singular entities that are under very tight genetic control. But in actuality, nothing could be further from the truth. We know empirically from anyone who's clinically treated these individuals that there are tremendous differences in a number of factors: the time and age of onset, the prognosis and progression in certain individuals who may progress more rapidly versus more slowly, and most importantly, in the actual response to various therapeutics within individuals that lie for virtually within any of these autoimmune disorders. Part of this heterogeneity is owing to the fact that there's different etiologies and different biological mechanisms by which an individual may go from a normal state to developing an autoimmune condition. Some of that is genetic, but the majority of it is non-genetic. And this is, of course, what gives rise to some of the heterogeneity. And the crux of precision-guided therapy and subtyping of autoimmune disease is really about being able to substratify these individuals and their disease processes. And the more discrete we can be in that substratification, the more closely we can align a singular individual with their disease and a particular therapeutic that's targeting their mechanism of action. And so this concept of autoimmune subtyping is certainly one, again, like precision-guided therapy, that is under constant evolution today. But I suspect, much like in the case of oncology, that this is soon going to become the norm, whereby we describe autoimmune disease less so by the actual tissue that's affected and much more so by the actual mechanisms that are marked by specific biomarkers that drive the disease process. And that's really what's going to allow us, as we can substratify these individuals and subphenotype these individuals, that's what's going to allow us to more effectively treat these individuals.
Great, thank you. So we've had another question come through, which is, in what phases of clinical studies would these biomarkers be most helpful for?
Yeah, thank you, Esther. So these biomarkers can actually be helpful at all phases of clinical development, from earlier phases like phase 1, all the way through phase 3 and even phase 4 post-marketing types of studies. The main difference, though, is that the questions that these biomarkers would address might be different. So at earlier stages, we might be looking for markers of pharmacodynamic and drug response, and at later stages, potentially for patient stratification. And at phase 3, we can even use these biomarkers that are discovered in earlier clinical phases to be able to stratify patients for enrollment, as well as identifying markers that could potentially be used as companion diagnostics.
Great, thank you. So the next question is, you mentioned your technology can detect broad chemistries, including metabolites and lipids, but can you give more detail about which particular chemical classes are detected?
Certainly. So our technology can capture a very broad, a wide breadth of chemistries. Everything from traditional polar metabolites such as amino acids, nucleic acids, traditional central carbon compounds, to polar lipids such as oxylipins, bile acids, endocannabinoids, all the way through non-polar neutral lipids such as di- and triacylglycerides. Now, traditional metabolomic and lipidomic technologies have relied on separate methods to capture each of these different types of chemical classes. And what we've been able to do here at Sapient is develop a very unique proprietary chromatographic technology that allows us to sample a very broad range of these chemistries and do that all in a single method or a single run, which allows us to have much greater throughput and capacity to analyze more samples and more molecules.
Great, thanks, Tanya. So the next question is, what is the process of going from discovery to translation of biomarkers? What is the status of the IRAE biomarker?
Sure, it's a great question, Esther. And this is very much a data-driven process, which is why, and the approach that we believe in. The high throughput technologies, the RLCMS technologies that Tanya alluded to during the seminar, are really meant as just step one in which we can use these technologies to measure thousands of biomarkers in anything from a dozen samples to over 100,000 samples. And then we let the data tell us what are the most important signals. And what we do is we sift through that data using advanced statistical approaches, everything from regression Bayesian statistics, all the way up through much more complex machine learning based approaches. And from those tens of thousands of biomarkers that are assayed, we can identify what are the most critical markers for the specific question under that's being posed. And then what we do is we advance those singular biomarkers to our high complexity CLIA CAP accredited laboratory, and we can rapidly develop the translational assay. In this way, we can go from R&D, measurement and screening of thousands of biomarkers, statistical analysis of data and prioritization, validation, and then rapid translation. And so this is, again, all driven by the underlying data. We don't surmise to understand what are the most important markers, but rather we go out and believe in measuring as many of these markers as possible and letting the data tell us what are the most critical ones for a particular biological question, whether it be understanding a diagnostic marker, again, prognosis over time or progression-based markers, or ultimately a therapy-based companion diagnostic. Now, with regards to the IRE biomarker that we shared with you during the seminars, you can imagine that's a marker that very rapidly went from initial discovery and as part of this process now is being translated. In this case, it's unique in that it's not only a potential diagnostic marker or prognostic marker that may reveal individuals who are at high risk of developing severe autoimmune events in the setting of immuno-oncology, but also the marker itself, as we showed in some causal data in the preclinical systems, also appears to be a very active component of the biological process that drives autoimmunity in the setting of immunotherapy. And so there's some very interesting therapeutic targeting that's occurring in that space as well. And we hope that in the very near future here, we'll have some updates with regards to both the clinical testing and the implementation and deployment of the clinical tests, as well as a status update on the therapy development that relates to this molecule.
Great, thanks, Mo. We've had another question come through. So the question is, the overlap in drug-induced autoimmunity and canonical autoimmune disease is interesting. Can you perhaps expand upon this idea of shared mechanisms with autoimmune disease states?
Yeah, absolutely. I'd be happy to, Esther. And so, as I mentioned, there's many mechanisms for any singular disease that may allow an individual to go from a normal state to what we would call an autoimmune disease state. And that's true whether or not it's RA, lupus, psoriasis, IBD, or drug-related autoimmunity. Now, as you can imagine, some of these pathways may be universal and shared across multiple diseases, whether they be induced, meaning a drug-induced autoimmune state, or a canonical lupus or IBD-based state. And some of these are unique to those specific disorders. So in the same way we can substratify individuals in a way that allows us to understand what population within a given disease may respond to a particular therapeutic, we can also similarly use the same approach to understand what other diseases a particular therapeutic may be beneficial in. And this is a way of extending indications. And certainly there's a very rich history of repurposing of therapeutics in the autoimmune space across various disease types. And this is certainly an area, once again, that using these precision or biomarker-driven approaches, I suspect is going to grow markedly here in the next decade.
Great, thanks, Mo. So the next question that's come through is, There's a tremendous amount of GWAS data available for autoimmune disease. What more can circulating biomarkers tell us about disease processes or drug development?
Perhaps I can expand upon this, Esther. So you're absolutely right. There's a tremendous amount of GWAS data in the autoimmune space, and certainly this has one of the victories, if you will, of the genomic revolution, in which we've learned quite a bit about the genetic landscape of autoimmunity. Now, the challenge has been in how we translate this GWAS information, this genetics data, to actual clinical practice, particularly for target identification, validation, and drug development, and that has proven to be not a trivial process. There's a number of ways in which biomarkers can help us do this translation or accelerate this translation. First, to begin with, for anyone who's peered into the GWAS literature and data, there's hundreds to now thousands of genetic variants and haplotypes that are associated with any particular autoimmune disease. And being able to organize these into pathways and shared biological mechanisms has been challenging. One way in which we can approach is by looking at the circulating markers that associate with these particular genetic variants and being able to cluster them according to shared circulating markers in a way that aligns these with particular biological pathways. The other approach in which we can leverage GWAS together with these circulating markers is through an integration analysis that allows us to identify circulating markers that read out specific genetic variants. And so as these genetic variants and their biological processes are targeted by various therapeutics, whether it be small molecule, large molecule, whether it be gene and cell therapies, et cetera, we can leverage these circulating markers as target engagement markers, pharmacodynamic markers, biological pathway activity markers in a manner that very quickly allows us to understand how drugs are working and in whom they're working. And so the way we think about it is that these small molecule biomarkers serve to complement GWAS and genetics and genomics, as well as proteomic type data sets. And this integration is where the real power of each of these modalities is then unleashed.
Thank you, Mo. So our next question is, can you please expand upon the human biology database, how this was assembled, and what type of information is within the database?
Yes, Esther, I'm happy to expand upon this. So our database is comprised of samples that have been collected from 30 to 40 different sources and studies. And these have mostly come through partnerships that we have with academic institutions and medical centers. So approximately 2/3 of the database comprises of samples that have come from individuals who are relatively healthy or normal. And over the course of time, approximately 10 to 30 years, they develop different types of diseases, or a number of them also remain healthy and well-functioning, which is also quite interesting to consider. And for these individuals, we have samples as well as longitudinal outcomes over time and phenotypic and clinical data regarding their health or disease status. The remaining third or so of the database comes from individuals with different types of diseases, from common diseases such as cardiovascular and metabolic, liver diseases, as well as individuals with very rare diseases as well. And these samples and individuals are very deeply phenotyped around that specific disease. Now, the database that we've talked about, it's a constantly growing asset or resource. We are adding samples to this database on the order of 50 to 100,000 samples per year. And it's something that we will continue to build and could even work with different partners and sponsors to build out in specific disease areas of interest.
Great, thanks, Tanya. I think we have time for about one more question. So the last one that's come through is, you briefly touched upon this, but can you further explain how Sapient works as a CRO?
Thanks, Esther. It's a great question. I'm happy to answer this one as well. So Sapient is a biomarker discovery organization. We do not do primary drug development ourselves. And we're a service organization that works not only like a traditional CRO, but we really see ourselves as a strategic partner with our sponsors in this discovery process. And so, we can provide fee-for-service projects, but as well as more collaborative partnership projects. And our deliverable is not only the data itself, but our goal is to really discover insights that can help drive forward and accelerate our sponsors' translational and clinical development programs.
Thank you, that's great. So that's about all we have time for today. So this draws the webinar to an end. Thank you very much for your questions and for your time today, especially to Mo and Tanya for your presentation. For further information regarding Oxford Global's biomarker series, please visit oxfordglobal.co.uk/biomarkers. All that remains to be said is have a great day, and we look forward to welcoming you to one of our future events. Thank you.
Thank you so much, Esther.
Thank you, Esther.